Well, hello everyone, and welcome to this special Facebook Live Ask the Stem Cell Team event about stem cell therapies and sickle cell disease. I'm Adonica Shaw, the Associate Director of Marketing Communications at CIRM. CIRM, for those of you who don't know, is California's Institute of Regenerative Medicine. What we do is we accelerate stem cell treatments to patients with unmet medical needs. Clearly, sickle cell disease is an unmet medical need affecting around 100,000 people in the United States, mostly African Americans, leading to lifelong complications and reduced life expectancy. So today, we're quite fortunate to be joined by three amazing people. Who are going to talk to us a little bit about the latest stem cell research targeting sickle cell disease and the challenges facing this approach. I'm going to introduce my guests in a second, but the first thing I want to say is that we're going to be as informative as we possibly can. We do value your time. We are going to get to questions. So if you want to email them to us, feel free to do so. But if you'd like to just type them out directly onto Facebook, our team will try to get them up on the screen and addressed. And anything we don't get to, we're going to try to put together a blog in the coming days and weeks if we have a lot of questions that just do not get tackled here in this hour. And of course, I want you to stick around to the end if you can, because we're going to give you some information on how to get to our clinical trials and some other things that you need to know before you sign up for any kind of trial or therapy. Okay, so our guests. I would like to introduce them in no particular order, but our first guest is Dr. Mark Walters, who's a pediatric hematologist oncologist from Children's Hospital Oakland Research Institute. Dr. Don Cohn, who is a professor in the microbiology, immunology, and molecular genetics department at UCLA. And Adrienne Shapiro, who is a patient advocate for sickle cell disease and the co-founder of Access Advocacy, sickle cell disease patient education and support website. Without further ado, I want to hand it off to Dr. Don Cohn, if you want to introduce yourself and tell us a little bit about your work.
Okay, well, thank you. As you heard, I'm a professor at UCLA. Besides being in the microbiology department, I'm also a pediatric bone marrow transplanter like Dr. Walters. And I've done research for more than 30 years on gene therapy. And for over the last 10 years, primarily funded by CIRM, we've done work on sickle cell disease, which has always been the big target, but it took probably a couple of decades for the techniques to get good enough to start applying to sickle cell. So I thought I would give a little background on what the approach is and a little of what we're doing, and then Dr. Walters could talk about some other work that he's doing both on his own and then also that we're doing collaboratively. So as you probably know, sickle cell disease is caused by an abnormality in the hemoglobin protein in red blood cells that makes the red blood cells be stiff and sickle shaped and block blood vessels. That leads to all the many problems. And the red blood cells that we have are made in the bone marrow from blood-forming or hematopoietic stem cells. So one of the treatments that really can cure a patient of sickle cell disease is to give them new stem cells that can make normal red blood cells that don't sickle. That's a process that's called bone marrow transplant or stem cell transplant. And Dr. Walters, who you'll hear from next, is really the world's expert on the use of stem cell transplant for sickle cell disease. To do a transplant from another person requires the person to be a really good match. So if they're an identical twin, they would also have sickle cell disease, but sometimes brothers and sisters can be perfect matches for the main tissue markers, and it can be accepted. But that's only about 10 or 15% of patients who have a matched brother or sister. And so a lot of the work in the field has been trying to find other donors, either unrelated people who are pretty good matches, or even using a parent who's only a half match. And those types of transplants have really been improving over the decades. And so the results, the results of these transplants have gotten better, but there's still risk. So to get the bone marrow from the other person to be accepted, the patient typically gets high dose chemotherapy like is used for cancer, but in this case to wipe out their bone marrow to make space for the cells coming in from the donor, and also to wipe out their immune system so they won't reject the cells. So gene therapy, as we're developing it, is really done in that context. So it is also being looked at as a bone marrow transplant, The major difference being, instead of the cells coming from another person, would be to take the sickle patient's own bone marrow stem cells, purify them in the laboratory, and genetically fix them so that when they're given back, they will make red blood cells that don't sickle. And this has only been happening in clinic in the last 3-4 years in just a small number of patients. So I just want to talk about sort of what is this genetic fix that we try and do. And there's sort of two main approaches that are looked at in gene therapy. One is to add to the stem cells a new copy of the gene. And this would either be a normal hemoglobin gene that could prevent sickling, or a modified version that acts like the fetal globin. So in blood cell production, before we're born, we make a certain type of hemoglobin called fetal hemoglobin that's good for carrying oxygen at the low level that a fetus is exposed to. After we're born, our stem cells switch over to making adult beta globin, adult hemoglobin, and that's where the sickle globin is expressed. So sickle cell doesn't onset that acts like the fetal globin. So in blood cell production, before we're born, we make I'm getting feedback of myself. Okay. And so it turns out that fetal globin actually prevents sickling of red blood cells. So there are rare people who inherit both sickle cell and a genetic change where they leave their fetal globin genes on, and they have much milder disease. And that was learned 25, 30 years ago. And so a lot of the therapies directed at sickle cell disease try and turn on that fetal globin gene. So for example, hydroxyurea, which many patients with sickle cell take and can have significant benefit, turns on that fetal globin gene and prevents sickling. So the gene therapy approaches that are really being studied all try and get that effect by either adding to the stem cells another copy of fetal globin or a beta globin gene that's like fetal, or they try and turn on the fetal globin gene. So everyone's trying to do that by different approaches, but to the same end of trying to turn on or make more fetal globin to prevent the red cells from sickling. So as I said, one approach is to add a copy either of the fetal globin gene that's on, or more currently or more recently, what people are looking at is actually trying to edit the genes in stem cells. And some of you may have heard of CRISPR, which is an enzyme that can be harnessed to actually specifically edit genes. And so there are efforts to either use CRISPR to turn on the fetal globin gene or to actually fix the mistake in the adult beta globin gene. And so those are the approaches, but all of them entail getting the stem cells from a patient with sickle cell disease, taking them to the laboratory, doing this genetic engineering trick, and then transplanting them back.
Okay. Thank you so much for introducing yourself and giving us a little bit about what you're going to be discussing with us today. Dr. Walters, do you want to hop in and tell us a little bit about your background and anything else you'd like to share about your research?
Yes, of course. And thanks for inviting me to be part of the discussion today. It's really, it's always exciting to talk about one's work. And I've been at this for a little more than 30 years now. I first became interested in sickle cell disease when I was in training, and I was in Seattle at the time. And that was a location where a lot of pioneering work was done in bone marrow transplantation. So this is a medical procedure where we use the blood stem cells from a healthy donor to replace the defect in the person receiving the transplant. And we thought a long time ago that approaching sickle cell disease in this manner might have a curative effect. So in 1991, I actually performed my first bone marrow transplantation in sickle cell disease. It was a 10-year-old girl who'd had a stroke, which was at that time was a fairly common complication of sickle cell disease. And we showed that after the treatment, that her healthy donor cells replaced the damaged sickle cells and protected her from having another stroke or any other complications of sickle cell disease. So since that first experience that I had, worldwide now more than 1,000 transplants have been shown, and they've really solidified the evidence that a bone marrow transplant using a brother or sister as the healthy donor is successful roughly 90% of the time. And I get challenged all the time, Dr. Walters, when are we getting a cure for sickle cell disease? Well, I think we established a cure back in 1991, but the community is not convinced, and for a very good reason. And that's because only 14% of the time will a child with sickle cell disease have a healthy brother or sister who can be a donor. And now we can look for unrelated donors who by chance have inherited the same transplant type as a person with sickle cell disease, but because African Americans are underrepresented in the group of volunteers who would be willing to give bone marrow if they were asked to do so for a person, we can find maybe another 19–20% of donors. So even if I wanted to treat every person affected by sickle cell disease who I thought should go through a bone marrow transplant, I could only treat about a third of the patients. So the gap today is the fact that we can't find donors for most of the patients, in my case, children, but also young adults who might benefit from a bone marrow transplant. So that's the first hurdle that we need to overcome. The second has to do with the fact that a bone marrow transplant is a really intensive medical treatment. It's done in the hospital, and the way it works is that we paralyze one's immune system for a period of time so that the incoming donor cells can begin to grow and replace the damaged sickle cells. Well, during that period of time, a couple of things can happen. A person can get a serious infection. The immune system of the donor can attack the recipient. That's a complication we call graft versus host disease. And in fact, that can be a fatal complication. In my experience, that's the most common fatal complication that occurs. So that's another important reason why a lot of families shy away from seeking a bone marrow transplant, because it's a risky procedure and you could die of the treatment itself. So the reason for the excitement is that we think we've found a way out of this conundrum. So we can use a person's own stem cells as the donor for this procedure that we're trying to develop. And because it's your own cells, there's no chance that you would get graft versus host disease. So right off the bat, we would eliminate this #1 problem that we face today. The second thing is that because everyone has stem cells in their own body, they can act as their own donor. So we overcome the donor problem. And what Dr. Cohen was introducing is this new system that was discovered not very long ago, 2012 or so, whereby we've harnessed something bacteria do to destroy viruses that they're infected with. So this is an enzyme that cuts the viral DNA up into little pieces so that it can't destroy the bacteria. And it knows the difference between the virus DNA and its own bacterial DNA because it's directed in a very specific way to a particular sequence in the virus, DNA sequence. So it's a little like a, your word processor program doing a search and replace procedure. So harnessing this system, it's now been tested in a variety of different organisms, including human cells. It's possible to use the word processing function to identify the target sequence, which in this case would be the Sickle mutation, a very unique sequence, happens only once in the human genome. Clip out the mutation and then edit back in the correct sequence so that you've now replaced the hemoglobin in that particular cell. And when we first started the work, Dr. Cohen was a pioneer in this. It was observed that the correction was only happening in actually fewer than 1% of the hematopoietic stem cells. So these are the mother of seed cells that would repopulate, replace the blood cells in the circulation eventually. So that's not nearly efficient enough to have a clinical benefit. We created a disease team. That's a group of scientists and doctors working together with funding from CIRM, which was terribly important to catalyze this cooperative research. and also involve teams in California that have expertise in all aspects of how one might bring this treatment to fruition, to the clinic to actually treat patients. So, so for example, Dr. Cohn's team at UCLA is expert in how to manipulate the stem cells in a manufacturing facility. There's a group of investigators at UC Berkeley that are expert in how to use these reagents to actually edit the DNA. Dr. Jacob Corman is a leader there. Dr. David Martin at our own research institute in Oakland is expert in how to test these human cells in a mouse model. So together, and then I bring to the table this ability to translate these advances to a clinical trial that we might one day test in real patients. So starting at less than 1% editing in that stem cell population, where we are today is in the 20 to 25% range. And we know from our experience in bone marrow transplantation that if you have just 20 to 25% healthy donor cells after a bone marrow transplant, that's a cure. So all the symptoms of sickle cell disease are eliminated. Those patients no longer go to the hospital for pain, for transfusions, for any of the other complications that are common in sickle cell disease. So we think we're very close to where we need to be to go to the next step. And in some ways, this is the most important step. Is this a safe treatment? So we still have a fair amount of work to do to demonstrate to our own satisfaction, as well as the regulatory agencies, that what we're doing with these blood stem cells is in fact safe to then introduce into humans who have sickle cell disease. So we're making good progress. We're especially grateful for the support that both CIRM, California Institute of Regenerative Medicine, and the federal government, the National Institutes of Health, have the resources they're now putting into these very exciting therapies so that we can, you know, in the next several years, in the next decade, certainly I would hope, have a curative therapy that might be really universally available.
Thank you so much. Adrienne, I want to get to you. I know that some of our viewers may know who you are, but I do want you to introduce yourself and tell us a little bit about your work with Access and a little bit about your advocacy work around sickle cell disease.
Okay, so I'm Adrienne Shapiro. A lot of you do know me. I'm a mom to Marissa and Casey. Marissa has sickle cell disease and Casey has trait. I have watched Dr. Cohn's work for over 30 years. He was the first person that I knew who was doing bone marrow transplants with sickle cell disease. And that's how I kind of, I guess, got my first footing in the stem cell and bone marrow arena. I started Access Advocacy because they had made so much progress with stem cells. And at that point, I realized the problems we had with finding the appropriate matches and the idea that using our own tissue to heal us was really the only way for us to have a universal cure. So I said, this is really great, and started an organization initially to get African Americans in the sickle cell community to understand and be a part of clinical trials. And as so I first dove into the stem cell world and figured out what they were doing and learned a lot. But I started to get calls from adults who, like my daughter, were running into trouble getting care. And I had no idea at that point that some of them were being treated so horribly, at least the majority of them. And so the focus of Access went from just being purely getting people into clinical trials and doing all the cool stuff to doing point of care advocacy, where we go out and we help those people who are navigating through the medical system. And that's anywhere in a clinical setting, most in the ER and and on the floor. Our goal is to support stem cell research until we get the universal cure, and to keep as many people alive as we can while they're doing that. and they're doing a great job of it. So, you know, I've had to speak to a lot of people and in front of a lot of people, and I've had to travel a lot. So I feel for those two guys who for years I watched speak in front of people and travel a lot. So not only are they in the lab working really hard, it takes a lot to support the science, and it takes a lot to support our community. And I think it's with their support, the support of CERM, the support now of the CDC, NIH, and ASH, sickle cell is finally under the spotlight. And I feel very, very optimistic about our future. And I'm happy to answer any questions anybody might have. Oh, yes, and I'm in an airport right now. So you may hear an announcement, or I might have to go on mute, but I'm so excited to be a part of this and so honored. So thank you.
Well, thank you. I just want to jump in really quickly. You know, thank you for joining us. If you're just now joining the live, this is a special Facebook Live about our panel here that's going to be talking about stem cell research and stem cell studies that are being offered and being currently developed for sickle cell disease. So if you have questions, please feel free to put them on the timeline. I see them on my end. And I think what we're going to do is we're going to start getting into some questions now if people are wanting to get their questions answered. I've seen several pop up here. And so I'm just going to go back down, and I'm going to put them up on the screen so that way everybody has the opportunity to kind of see some of the questions that I'm getting. And so this might be a question for Dr. Walters and Dr. Cohn, and it comes from Cammie Gallagher, and she's asking, if we were able to use embryonic stem cells, would we be able to treat all of them? I don't know if one of you wants to jump in to tackle this particular question or to address it, but that's one of the questions that we've had pop up recently.
Yeah.
Oh, go ahead, Mark.
Oh, I was just going to say, I think that's an interesting question, and it's certainly one that a lot of scientists have thought about. There's also a way to create an embryonic stem cell-like cell called an induced pluripotent stem cell. In these disorders, the hematopoietic stem cell is actually an adult type stem cell, and what's nice about that approach is that we know that adult type stem cell will turn into a red blood cell that'll be useful in the blood circulation. The challenge in the embryonic type of stem cells is that while you can direct them to become a very early progenitor blood cell, that is one that could give rise to a white blood cell to fight infection or a red blood cell to carry oxygen, it's very difficult to get those blood cells to behave like they would in the bloodstream, like a mature blood cell doing its job that it was meant to do. They have a tendency to remain immature. So that, unfortunately, hasn't been a universal approach just yet. There are still some problems that we need to overcome.
Okay. Do you have anything to add, Don?
Yeah, I mean, I agree with what Mark said. The induced pluripotent stem cells that he referred to, which are like embryonic stem cells, can be made from each person. And so therefore they could also be made as a perfect match. And so I think, you know, the question, could this be used widely? The embryonic ones don't match. They're only from a specific person. But if you made these induced pluripotent stem cells, they could be made from the individual patient. But as Mark said, it's really, we have a technical hurdle to turn them into real hematopoietic blood-forming stem cells before we can use those. So I think that's maybe 5-10 years further into the future.
Okay, so I do have another question for you, Dr. Cohn. So I know we've seen some success with sickle cell disease using stem cell transplants from bone marrow, but why do we need to try additional approaches?
Well, I think the answer is the point that Dr. Walters raised, is the absence of matched donors and the immune reactions. And so this would be another type of bone marrow transplant. It would be the patient's own. And so that would give everyone a donor and would give everyone a perfectly matched donor. And so in theory, if the gene therapy part is very efficient, it should be better than a transplant from another person. That remains to be shown, and some years off before we really know that. But the theory is that because it's the patient's own cells, it should be a better type of bone marrow transplant.
Let's see. And this question is open to Dr. Cohn or Dr. Walters. But are there any side effects that people should be worried about by approaching some of these other types of therapies?
I'll let Dr. Cohn start that one.
Okay. Yes, definitely. I mean, Dr. Walters referred to it, but this is a major medical procedure to have a bone marrow transplant, whether it's from another person or from yourself. And it typically entails a month or more in the hospital. Before the cells are given, and the patients are given typically high-dose chemotherapy to make space in their bone marrow. And so there are potential complications from the chemotherapy, damage to organs, risks of infections. And so it's nothing to be taken lightly. But as we've gotten more experience with doing transplants over the last several decades now, the risks have decreased. But there are definitely risks. And then one other topic that we didn't touch on, these chemotherapy drugs, one of their side effects could be to decrease fertility, maybe even to cause sterility. So that's a major consideration for people who are planning to have children. A bad side effect of the treatment could be inability to parent children directly.
And I just want to say that in my discussions with our community members, that's one of the things that comes up quite a bit, is the fact that many of them would like to have children and feel that, this will rob them of that right or that ability. And so the choice is, do I live with the devil I know, or risk never ever having children? And so, and of course, we have had several people who've done bone marrow transplants and they've failed, and getting people again to sign up and go through that whole process for donating and receiving is a very difficult thing. So whatever can mitigate the rejection rate or the complication rate. So that's why we think of stem cells as being the universal cure. We'll all be our own donors.
Now, another question that's come up, I'm going to show it here on the screen so that we can all see it together. Could any of the stem cell research on sickle cell help with other blood diseases or disease in general? Does anybody have any particular feeling or sentiment they'd like to share to address Mr. Klein's question?
Yeah, I can jump in. So the answer is yes. There are a large number of hereditary blood diseases, and all of these have an origin in the DNA. So that if you had a way of adding a healthy copy of the defective gene in the way that Dr. Cohn described, or if you had a way of targeting a small mutation in a particular gene by the CRISPR-Cas9 reagents, those other diseases also should be treatable using the same approach. So a lot of us view sickle cell disease and a related blood disorder, another hemoglobin disorder, thalassemia, as the proof principle. And if these are successful, then we might begin then to look at other rarer diseases. So even though sickle cell disease and hemoglobin diseases in general are rare in this country, there are many more far rarer disorders. And because this approach is quite personalized, you have to discover the mutation in each individual and then tailor the treatment around that particular mutation in that particular gene. You can see why you almost need a catalog approach to develop each of these. So it's time consuming and expensive, but absolutely there's no theoretical reason why something that works in sickle cell disease couldn't be applied to other hereditary blood disorders.
And there's probably about a dozen or more other diseases that are being treated by the same type of treatment: gene therapy, for a number of immune deficiencies. We've done a lot of work with bubble babies, kids born without immune systems, giving them back into their stem cells the gene that they're missing. We've treated some other white cell diseases. So there's probably 30 or 40 different diseases that affect blood cells that can be treated by this approach. And so what we learn for those diseases helps us treat sickle cell, and what we learn in treating sickle cell helps us treat those other diseases.
So I have a question. What about blood cord banking? Is that something that people can use? Is that a viable option to kind of address some of the concerns of parents who are worried that they might pass along, for example, the sickle cell disease gene to their children? Is that something or another avenue that people can take?
I'll jump in there too. So yeah, I'm a big proponent of families affected by sickle cell disease and other blood disorders making arrangements to collect the cord blood of a sibling or a child who's at risk. And there are several national cord blood banks that that do that at no cost to the families, at least the collection part. I think my dream of dreams would be to collect the cord blood from an affected infant at the time they're born, do this editing right away, and then reintroduce those cord blood stem cells before the child begins to have problems related to the underlying sickle cell disease. Now, we're a very long way from being able to do that, but I think it's important to set goals that are very lofty, and to begin to find ways to do this safely in a very young child, so that we could really end the disease before it gets started. So having a number of families who do collect the cord blood, having those cord bloods in a bank that are accessible for use in the future, and also for research to help us develop this approach, I strongly endorse.
Mm-hmm. Adrienne, do you have something you would like to add?
Well, we did that with Casey. She calls herself the spare. She's not on the call now, but and they were half siblings, so it didn't work because of where Marissa was, particularly in the disease. But we're really encouraging all mothers and fathers and families to do exactly that, to save the cord blood. To me, my ideal would be that everybody does it, right? And makes the material available for research, available for what's going to happen in the future. I think.
Are there programs available for parents to do that reasonably easy in a way that's cost effective?
Well, yes. I can't give you any, but we will give you the list of those. But yes, there are. And having gone through it, I mean, you get rid of all that stuff anyway when you have the baby. And close your ears. So it really is not, it's a very simple process. The medical staff handles all of it. And, it's like, to me, one of the sort of easiest but most important things I think we can do in terms of looking out for the health of our loved ones and basically for everyone.
So I have a question that's popped up for Dr. Walters, and it comes from Shirley Davalos, excuse me. She's wanting to know when your work is going to be ready.
Yeah, well, that's a very frequent question we get. We are working as fast as we can, but I have to circle back again to the issue of safety. So this has never really been applied yet in humans, where we're to be very careful. There are some concerns that, in addition to modifying and correcting the sickle mutation, there may be similar areas in a person's DNA sequences and in other chromosomes, not the one that the sickle cell gene is located on, that also get altered, or that we bring genes together accidentally that aren't meant to be together that could, after the fact, say, promote leukemia, which is a blood cancer that develops from the blood stem cell. So we need to be as certain as we can about making sure that we don't do that. So that takes time and some pretty labor-intensive study. So so we think we're at least 1.5 to 2 years to even doing a very early stage study in adults with sickle cell disease, and that's really optimistic. If we run into problems, and let's face it, clinical trials, the development often have problems that you don't foresee, and they have to be dealt with. So it could be longer. It could be 2 to 3 to 5 years before we begin. But I don't think it'll be longer than 10 years. That's for sure. So I think we're getting close, but we still have more work to do.
So when it is eligible, who's going to be eligible for it if it does get or becomes available within the next year or so? If you're lucky enough to have it pass that quickly.
Well, whenever you're starting a new study and a new treatment, you haven't proved yet in anybody that it's beneficial. So so you have to target individuals who are having a lot of trouble related to the sickle cell disease, and they have to be adults who can give permission, consent to be in a clinical trial after they've understood all of the risks involved. So they tend to be very small, not more than 10 patients, and generally, quite ill patients, but still healthy enough to go through an intensive treatment. So it's pretty selective at the start, and then if in fact we can show in the first 5 to 10 patients that it looks like it's having some beneficial effect, and it appears to be safe, at least in the 2 years after a person has received the treatment, that's when we begin to open it up to a broader array of individuals, and maybe also children whose parents can give permission to be in a clinical trial.
Okay.
So let me just comment that, so Mark is talking about the approach of using the gene editing. There are actually currently several clinical trials open across the country using sort of the more classical approach of adding a gene to stem cells. We have one at UCLA, there's one at Children's Hospital in Cincinnati, and there's a company, Bluebird Bio, that Mark has been part of that has treated several patients. There have been a couple of patients that have had very good responses with that approach. And so in fact, there are several different ways to do it. We don't know what's the best one, but there's certainly beginning evidence that it can be effective and really have the same benefits as a transplant from a healthy donor can.
We have a question that popped up while we were talking. I'm going to show this. It's again from Cammie Gallagher. She said they saved her son's cord blood, but wouldn't his cord blood stem cells be predisposed to his ADHD? Dr. Walters, Dr. Cohn, do you have any sentiment around this question or any feelings that you'd like to, I guess, express for Ms. Gallagher?
I'll address this one. So the reason we're talking about cord blood is because at the time a baby's born, on their umbilical cord blood, there are stem cells like we get from bone marrow later. And if they're transplanted, they can affect blood cell production. They wouldn't affect a behavioral or a neurologic condition like ADHD. So I think they're really somewhat unrelated, and it wouldn't affect it either positive or negatively.
Okay. So this next question is going to be for Adrienne. It comes from Marissa Coors. She's wanting to know what the sickle cell community can do to support this kind of research. Adrienne?
Yes.
There we go. I didn't hear you for a second. So we have a question from Marissa Coors. She's wanting to know what can the stem cell community do, excuse me, the sickle cell community do to support this kind of research?
Well, I mean, we can and are, of course, going about educating the community. We are speaking with legislation. We are watching what's happening to make sure that once the research gets proven and that people can get paid and we can have access to it, we're volunteering. But are we doing everything? I'm asked the doc. So those are the things we're doing now. We're now thinking ahead of the game. We're thinking, you guys are doing very involved work, very expensive work. You're doing great science. I mean, I'm so proud of it. I'm so proud of the quality of science that we're doing here. What else can we do to help? From
well, I know you're very involved also in efforts to educate physicians across the country, yes, because although there are a few experts at some centers familiar with sickle cell disease, so many patients get their medical care not in that setting, right, in the community clinic, in an emergency room. So getting the word out to them about best practices of managing pain for patients, it's such a unique medical situation, and we need to educate physicians and nurses across the country so that all the, just for separate from gene therapy and transplants, just so that their day-to-day care is the best possible, because I think that makes a huge difference in quality of life for someone. And so I know your efforts under that, I think, are really very important.
Well, we're just in a hurry. I shouldn't be laughing, but there's nothing you guys haven't heard from me before. Yeah, we are. Now the NIH, what is it? I always get it confused. NIH Blood Lung Institute, right? Mm-hmm. Has just announced where they're seeing a cure for sickle cell within the next 5 years, and they're planning on this big launch next year. And I know that, Dr. Cohn, your study is going out for the next, 3 to 4 more years, and also yours, Mark. Are you guys all working together in that, or are you explain to us how that kind of works? Because I think it's very unique.
Well, so we have a trial ongoing now at UCLA, just adding a gene to stem cells that we're doing ourselves, that we started and that we're doing. But we have the work that Mark was talking about, the gene editing, is a multi-group collaboration. And so Mark is our leader. As he said, people from UC Berkeley who developed this CRISPR technology, in my group that has the experience with processing stem cells in gene therapy patients. We've put our forces together now for the last 3 years, I guess it's been, to try and bring this to the clinic. And at the, beyond us then, there are many opportunities in which we all get together as scientists and compare our results, present our results, present our findings. And so, we want to present it first, but then we want to share it. And so I think there really is good sharing of best practices across the field.
If I could circle back to the sickle cell community question about getting involved, I really want to reinforce what Adrienne is talking about, activism. It's really contacting congressmen and congresswomen and exerting political action to raise awareness about sickle cell disease and the fact that so many of our patients and families in the community are underserved. They don't have access to the kind of expertise they absolutely need to have a successful outcome to address their healthcare needs, and it isn't right. And I think that begins at a grassroots level, and it begins by making awareness about how. Just start with access to competent healthcare that's culturally competent and delivered without barriers or preconceptions about what might be causing pain in an individual, instead of just believing that the pain that they have is important and should be treated. And then it mushrooms out to the curative therapies that we're working on. Another thing we're keenly aware of is the cost of these new therapies, the stem cell transplantation techniques that we're talking about. If it's gene therapy, because in the early days that's going to be incredibly expensive, and will payers be willing to cover these curative therapies? Will the cost be prohibitive? And what is our federal government going to do to ensure that patients and families have access to the new treatments that have so much promise? It's a healthcare policy issue as much as it is a scientific, physician problem and issue. So I would encourage the community to begin now think about those potential problems and to help us find solutions to them.
Thank you so much, Mark. Adrienne, somebody sent a notification. So another question from Shirley Davalos. She's wanting to know how people can get involved with your group.
Oh, just send us an email or call us, 323-506-2224.
Thank you so much, Adrienne.
I just wanted to say that I have been in this battle all my life, having had a brother who had sickle cell disease. But I am truly, truly, I don't even know if hopeful is the word. I see now that in these meetings where there used to be just us patients trying to find someone in the policy arena to hear our plea for help, that we were actually last month in a meeting where we had pretty much the players from the payees to the top federal CMS, NIH, CDC, all together. And of course, we brought up this subject of who's going to pay for it. And for people who have been marginalized culturally, we have learned not to expect much. But they're already at this point looking at that, and I feel very optimistic. And I think we have to be prepared to support that. We have to be aware of how much money we cost to keep ourselves healthy and alive. I know that, sorry about that. United Airlines. I know that a lot of times we aren't tracking it because the government's paying for it. But I think it's more, that makes it even more important for us to track it so we can talk value. So it may sound like a lot of money. Let's say, we're there, and it's going to be $250,000 to $1 million to treat one person. Well, compared to a lifetime of hospitalization, and the lack of ability for that, whoever that family member is who's supporting them to work and pay taxes and buy things, really for the government, it becomes really cheaper in the long run to pay for it. But we've got to have the numbers. We've got to come in prepared to have those discussions. And we've got to show up and talk to people about it. And one of the things we're trying to do is make sure that everybody knows who's having these discussions and when they're happening, because a lot of this has always been done in a vacuum. And I'd have to say that now, again, and I'm going to give props to CIRM for doing things differently, for doing their stem cell their trials, medical trials differently by including stakeholders who have the disease and are living with the disease, and giving us a voice and support, and supporting us even when we're going out and just trying to figure out who to talk to, because we don't know who. There are national organizations, and a lot of times they're working in a vacuum, and they have a thing that they do and their people. But for those of us with boots on the ground just trying to stay alive, it's pretty lofty to think, I'm going to walk into a meeting just as a mom and talk to the FDA or the CDC or, NIH or the Medicaid people, or, because our dealings with them of trying to get service is so difficult. So the idea of going to them to try to get any type of higher level access seems impossible. So we can get that, and again, we're going to try to get word out to everybody. But that's what I think we have to be doing now.
Thank you, Adrienne. If you're just now joining us, we are moving along here. I know that we've got about 10 more minutes, but if you do have questions, please go ahead and type them onto the timeline, and we're going to try to get through as many of them as possible. But anything we do not address, again, we're going to try to put together a blog in the coming days, in the coming weeks, to address some of your questions and concerns. Jumping to the timeline, it looks like we've got a question from Jeanette DeJesus, and she's asking if we can mention the locations and states where gene editing is taking place. And she wants to know so that she can talk about it with her son's hematologist.
Well, do you want me to talk about it? If you want to find out, there is a place online. You can go to the government, it's clinicaltrials.gov, and it'll give you a listing of all the clinical trials currently going on. And they list not only what's happening in gene editing, but we also have some exciting trials going on now for treatments. There are several new meds that are currently in stage 2 and 3 trials. And you can go there. CIRM, I think, has things listed, or they probably have a link to the government site also. Or you can contact us at Access Advocacy.
Yeah, I would just chime in that there is a network of California medical institutions called the Alpha Stem Cell Clinics. There are now 5 funded centers in Northern and Southern California, and CIRM lists on their website under the Alpha Stem Cell Clinic heading all of the clinical trials that are open and enrolling patients statewide. So that's another place to look, and would include for example, Dr. Cohen's trial in sickle cell disease, and also my own trials that are currently enrolling patients in gene therapy.
And I just want to say, I don't know where you're listening from, but if you're not in California, don't discount it. Don't ignore a trial that's going on. If there's something you're interested in and you think may help you or your family member, follow up on it. Give them a call, and don't give up. Just
yeah.
Show up.
I would echo what Adrienne just said. I would say with regard to participation in these gene therapy and gene editing trials, I see as many patients from outside the state of California as I see in California. So if you can travel, that's great. If you can't, we often do these by telephone just to get some educational issues and topics discussed. I'm happy to do that. So don't let geography be a barrier to finding out about these exciting new therapies.
And I just want to say that sorry, talking too much again.
No.
That one of the things that I'm currently doing now is that I'm actually a CAP member on 2 of the trials, the stem cell trials being funded through CIRM. And it has given me such insight into what's really involved when it comes to clinical trials. So you really have to understand that the people that are working in the research have our best interest in mind. It is to all their interest to do good science, to provide a place where you as the clinical trial subject are supported and looked after well. I never thought I would have a voice at a place like that as a mother, but they've welcomed me. They listened to my ideas. They've implemented things where it makes sense to implement them, and it's been a great, I don't know, it's been an honor. Mm-hmm. But again, I just want to say, good science is expensive science, and it needs all of us. So.
And if I could just amplify on that. So
I think we might have lost Dr. Cohn there, so I'm assuming he'll.
Can you hear me? Hello?
Pop back in.
I can hear you.
Okay.
When he's been able to maybe reboot on his end, but I don't see him on the screen, and I apologize, but if he pops back in, then you'll see him in that lower quadrant. We do have another question here, and this one might be something you might want to tackle, Dr. Walters. Um, it comes from Danny Keller. He asks, how do we keep up with other countries that don't have ethical, as in non-embryonic stem cell research?
Yeah, another good topic. I don't know if I can do it justice in 3 minutes, but I hearken back again to this alpha stem cell clinic that's approach that CIRM has founded and now expanded, because it's really, I think, a model for how to do cutting edge stem cell therapy. So it's high quality, safety is at the forefront, and it's done by teams that are experienced in conducting these kinds of trials. And for that reason, I think too, it's become a magnet for industry to where there's a need to quickly get a clinical trial up and running and to get some information about whether or not it might be useful. The Alpha Stem Cell Clinic seems to be the right destination for those folks as well. So it's easier to describe how to do it correctly than how not to do it.
Yeah.
But I think that's what you're getting at, is how, it really should be done ethically. It should be done with informed consent, with high regard for safety, and but also with an eye to accelerating the work, so that we don't get bogged down in the regulatory quagmires, and that we have skilled people that can navigate those systems, so that we can get the trials running efficiently. So so that's really how I'm going to end, I think.
Okay. Well, sadly, we've run out of time. I'd like to thank our guests, Dr. Cohn, Dr. Walters, and of course, Adrienne Shapiro. I want to, let you know if you're interested in being a part in any of our clinical trials, and I know that we've talked a little bit about it today. Please go to the CIRM website at www.cirm.ca.gov. I believe we've actually put the link to the page in the comment section if you'd like to find it pretty easily. And if you go to that page, you're going to find all kinds of good information that you're going to need to get questions about trials, and to make sure that you are getting into a scientifically rigorous program. You'll also learn about red flags to watch out for and help you steer away from any type of questionable program or something that's not being done ethically. And of course, if you have any questions for the stem cell team over at CIRM, please feel free to email us at info@cirm.ca.gov. And again, just to make sure you have it, that's info@cirm.ca.gov. And for all the latest and greatest stem cell research and information about all the latest at CIRM, please feel free to check out our Facebook page, go to our Twitter page, and of course, the Stem Celler, our blog. I myself actually put blogs up on that blog. So if you're wanting to know what we are looking at, some things that are coming out that we're interested in learning more about and sharing with you, please head over to our blog. Thank you so much for watching and for participating with us for another round of Ask the Stem Cell Team, and we hope to see you next month.